हेलो, हाँ भैया, अभी शुरू हो गया होगा ना? ठीक है, तो अभी
मैडम आप लाइव हो, यू कैन स्टार्ट।
म्यूट करके म्यूट नहीं म्यूट ही कर दो। और पहले मैम इंट्रोडक्शन आप सीधे बोलो लॉगिन ठीक है।
मैडम आप लाइव हो,
हाँ जी,
ठीक है, ठीक है।
स्टार्ट करें सर।
यस प्लीज।
A very warm good morning to one and all present in this lecture. I respect I respect Dr. Girish Sharma, professor in the Department of Fruit Science at Dr. Y.S. Parmar University of Horticulture and Forestry, Solan, faculty of the Department of Horticulture, and my dear students. It is indeed a very pleasurable moment to welcome Dr. Girish Sharma this morning for the lecture on the topic breeding methods in fruit crops. I feel honored to greet you here today at the Dolphin Institute, Department of Horticulture, to deliberate an important concept of horticulture in the breeding methods of fruit crops. Uh, Before proceeding for the lecture of Dr. Giri Sharma, I would like to give a brief introduction. Sir has over 29 years of teaching experience at undergraduate and postgraduate levels. Sir has guided 22 students, 6 PhD and 16 MSc students. He has delivered lectures in specialized trainings, TV talks, field demonstrations, farmer scientist interaction, etc. for the last 28 years. He has established and developed new fruit crop block of strawberry, macadamia, new apple scab resistant advanced seedling blocks, plum and apple hybrid testing blocks. He has evaluated the entire fruiting germplasm of apple for different pomological traits. Sir has handled more than 14 projects funded by different national and state funding agencies. He has over 95 research publications, 20 book chapters, 10 popular articles, and one book. Sir has performed new germplasm blocks of apple and pears, which were established, maintained, and their clonal rootstocks were multiplied. Plants of tree tomato, jamun, passion fruit, and raspberry have been raised. Besides this, germplasm of strawberry, olives, macadamia, persimmon, kiwi fruit, passion fruit, wild pomegranate, caronda is also maintained. Evaluation on wild pomegranate to isolate superior genotypes suitable for anardana making and breeding new cultivars in Solan and Sirmaur region. Sir has also worked extensively on day neutral plants, strawberries. He has evaluated different strain species and identified Clementine denule, Valencia late, Dholakua seedless as better strains. He has worked on strawberry evaluation and characterization with molecular and morphological markers, RAPD and ISSR. So this was all about a brief introduction of Dr. Girish Sharma. Now I would like to request Sir to deliver the lecture on the topic breeding methods in fruit crops. Please, sir.
Thank you, Dr. Kanika. I am
Thank
extremely happy that an opportunity has been provided to deliver a lecture on my subject. And before I start the actual deliberation of the given topic, I would like to know the status of my audience. Are they BSc students or postgraduate students? Hello? Hello? Hello, Dr. Kanika?
BSc student बोल दीजिए।
Sir, hello.
Yes, madam.
Hello.
Yes,
Sir, these
madam.
are UG students, UG students, sir. BSc students.
Okay. Okay. Fine, madam. Fine, madam.
Okay.
Uh,
Okay.
now I start with the topic that is breeding methods in fruit crops. A subject itself is very important because several governments, our governments, they have emphasized the importance of fruit culture in India after Green Revolution, uh, keeping in view the huge uh, population. It is very desirable that equal attention be made to the cultivation of fruit crops, as they hold several ingredients which are not present in the normal cereal crops. To begin with the topic, a small introduction: how the fruit culture advanced from nomadic to the present day, from Stone Age to the modern science. Almost about 10,000 years ago, during the Neolithic Revolution, agriculture process started. Though not in a scientific way, but still the process started. Thereafter, from 8,000 to 5,000, the second revolution took place, and here the villages to urban communities started developing. Thereby, during the Bronze Age, horticulture, irrigation, and other aspects of horticulture, they were refined, and people started to cultivate both agricultural and fruit crops in a much better way. Thereafter, from 3000 to 2000, Greeks and Romans did remarkable work in the cultivation of fruit crops. It is important to know how the fruit crops have evolved and where they have evolved. As a student of undergraduate, uh, you must have studied Darwin's theory and related aspects. Then it is important that how several fruit crops all around the world they originated, and when migration routes were somewhat simple, these crops were taken by missionaries, invaders from one place to another. They planted at their own places, and thereby dissemination of fruit crops from their place of origin moved to the other places where they were not present. The Mediterranean basin comprises of date palm, fig, grape, olive, pomegranate. In Europe, the temperate fruit crops. So is in the case of Central Asia. In East Asia, which we belong, has huge diversity in the form of several crops already indicated in the presentation. Then Africa, fig, coffee, Americas, avocado, blackberry, papaya, pineapple, and so many other crops. Here, Americas is referred in totality, taking into account the North, South, and Central America, whereas South America and Mexico, they have high diversity of tropical, subtropical fruits, and America, USA, alone does now in present diversity in case of pecan, walnut, and the blackberries and related crops. Further, it is important to know how the domestication process started. First was the recognition of the species. It was followed by selection of elite genotype. Thereafter, once it was established that they are of direct use to humankind, their vegetative propagation technology or techniques, they were finally refined and they are now in use. Then cultivation technology, thereafter, once there was sufficient plant material available, then came the cultivation technology regarding pruning, training, irrigation, pollination, and so many other related factors. And finally, the utilization of the ultimate product after they have been sold for the consumers, then its storage, drying, fermentation, processing, uh, these all uh, factors provide large income to the cultivators. Then the dispersal of fruit crops with human migration, as already said earlier, several fruit crops, there are incidences that crop was not available in one particular place and now it is growing in a very large quantity. Example is of guava, which is indigenous to South America, and now presently in India it is cultivated to such an extent that it is difficult to believe that this crop is not Indian crop but has been introduced. Such is the importance of acclimatization of uh, crops, and this happens only when uh, climatic conditions are equivalent to those that prevailed in its place of origin. And finally, the most fruit crops are close to the wild species than annual crops. This is important because uh, fruit crops, they are usually propagated asexually, and there are less chances of uh, changes. Once they are hybridized in nature, ultimately they cause variation. Then these things, they are important from breeding point of view, and some major problems associated with breeding, they comprise of long generation cycles, 2 to 10 years, depending upon the species in question as well as the varieties taken. Some have short period of juvenile phase, and others have very long. And so selection and development of horticultural fruit crop varieties, they are somewhat difficult compared to the cereal crops. Most of the fruit crop species, they are highly heterogeneous and require large population for effective selection. Number of species are polyploid and polyembryonic in nature. They also hinder the fruit breeding process. Then presence of parthenocarpy, of course, required, but from breeding point of view, it would be, and it is considered as a negative uh, trait, because very few seeds are obtained, and since the progeny is very limited, selection becomes very difficult. Now, there are specific breeding objectives, and especially for two categories. One is for rootstock, and another one is for the sign variety crops, sign varieties of various crops. And these related parameters uh, vary from crop to crop and species to species, and much work has been done for the improvement of sign cultivars and relatively less for rootstock. Now there is a feeling that is to promote and advance horticulture. There is need to pay equal attention to improvement of rootstocks, because climatic conditions are changing very rapidly, and there is need that rootstocks which are dwarf in nature, which are highly adaptable to marginal soil conditions and have long living time, lifetime, so that the sign variety remains healthy and free of number of insect, pest and disease attack, so that farmers harvest rich dividends from their plantations. As said earlier, sign varieties are equally breeding objectives of signs are equally important. Now, at earlier times, whatsoever was the material introduced, if we take example of temperate fruits, entire industry of horticulture is based on introductions from different parts of the world. Since they acclimatize to the conditions of India, uh, these fruits are being cultivated, and in Himachal Pradesh has established itself as a huge industry, right from the plantation to its end product use. So now need is that more plants be planted in a smaller area. The sign varieties should be precautious, prolific bearing, high productivity and quality, resistance to biotic and abiotic stresses, attractive fruit color, as pleasant aroma, processing and export qualities, good keeping and transport qualities. Now, importance of fruit breeding cannot be ignored, and from very early times, say the Vedic times in India, uh, though the actual principles operating in improvement were not known, but these improvement work was being carried out in the nature, as well as the inquisitive people who found these changes. They somehow got this material propagated, and diversity was enhanced. Now, the major aspect is of the day's talk is breeding methods for fruit crop improvement. First is introduction. This method comprises of introduction of plant material from a place or a fruit type which is not present locally. This process was earlier done by the missionaries or the invaders. They carried their food material from their place of origin and transported it in plenty to the places where they wanted to conquer and live there. Example is the Western way from Iraq, Iran. Mughals brought walnut and other fruit crops which were in plenty at their places, and during their travels they ate the fruit, and some was left behind. That material somehow uh, got germinated, and diversity from their native place was brought to several places where this plant material uh, was left out for any reason. And now, presently, that was a raw material of introduction, but now, presently, very precise ways are have been found out. That is, the place where introduction has to be made and from where it is to be made, they are they should match climatically. Reason being, any plant can grow at anywhere, at any place, but what is the end product needed is, if it is fruit, then naturally the production out of it. And for that very reason, adaptability is the most important aspect and must be given due importance when such material are introduced in India. Example is the delicious group introduced to India from California. Though English varieties were introduced much earlier and they were planted in Kulu Valley, uh, they did fruit. Their taste did not match the liking of the local people. They were somewhat acidic tart, and these varieties were not accepted by the local people. But the delicious group, being sweet, red colored, matched the taste of the local people, and in a short period, a huge industry got developed. This is the first way how the breeding method is, was, is, and would continue to enhance horticultural activities. Now comes the second method of breeding, selection. This comprises of bringing or making selection, whether from it is introduction. What happens is, during selection, it is not one or two varieties. Several varieties are brought from different countries. They are planted at specific stations for their performance, and thereafter, the ones who are performing better than the already established cultivars, they are selected, and these are multiplied and given to the farmers for cultivation. The second way of selection is the is. Say, example, in India itself, there is huge diversity of several tropical and subtropical fruits. So there is no need to introduce cultivars from any other place. Selection has to be the first criteria in improvement. Select, make a surveys, explore the areas thoroughly, get acclimatized the various variations present in the crop. A quantum of variation in characterization would ultimately help to select some desirable types which later on can be multiplied and they would fulfill the needs of local farmers as well as the better ones for export purposes. Third method of crop improvement refers to hybridization, and once you have built up a respectable germplasm via introduction or through local selections, then it becomes imperative that hybridization process is followed. In this method, what is done is two parents. One would act as a donor and other as the recipient, the female one. Then be based on the characterization, as said in the selection or introduction process, suitable types are selected as donor and recipients, and then they are crossed with each other, and the resultant seed is collected and then it is grown. And as per the objectives, the seedlings are evaluated, and once some suitable character has been found, then again they are mass multiplied, and after sufficient testing they are released as an improved variety for for a definite character, and it adds to the germplasm and thereby enhances the variability. Entire gamut of crop improvement rests on the variability that is prevailing, that is present in the form of material introduced or local selections, and then the third through hybridization. Sometimes what happens is all the three methods are not producing the required objectives. Then random method is mutation. This science developed when uh, Madame Curie found out the potent chemical radium, and thereafter, being very penetrating in nature, it was thought that it would help in bringing about changes in the genotype of the uh, plant material used for mutation work. And in the initial stages, variation was obtained, but what was the ultimate result of if it is apple for more than a century? Testing revolved around the treatment of various germplasm cultivars, species, and then thereafter the variation found out was categorized. This variation was not stable and regularly it occurred, and then the impact was lost, sometimes lost forever, and sometimes it reappeared and made the selection by the breeders very difficult job. Now, this mutation science in combination with hybridization, maybe in vitro or by other methods, is paying dividends, and several new forms are being evolved. Major difficulty in mutation building is the occurrence of chimeras, that is, single organ may comprise of two variant cells, then their expression is not stable and keeps on changing. It takes a lot of time for their stability. Uh, they are useful, these chimeras, they are useful. Their variation is required for, say, in ornamental crops, variation in flower color, extremely important. And for these crops, chimeras are important, but for main horticultural crops, they are negative, and now through biotechnological techniques, these chimeras they can are easily seen at early stages, and they can be rocked out from the further multiplication. Following this difficult path is the polyploidy, again very tedious job, and these breeding improvement methods have been placed in chronological order. That is, the simplest one, the first one, and as we go downward, it becomes difficult. Then polyploidy again, uh, in principle, very close to mutation breeding work, and the most potent chemical for polyploidy induction is colchicine, a very toxic poisonous chemical, and major impact of this chemical is inhibition of spindle fiber formation. Once it is hindered, then reduction of chromosome does not take place, and thereafter uh, progeny obtained is at a higher ploidy level. This ploidy phenomena in crop breeding is essential, especially where the individual fruit size is small. And in horticultural crops, there are several fruit crops which are small in size. In the cases of rubus, strawberry, and several other fruit crops where edible part is very small, and to enhance or to increase edible part, polity does or may play a significant role uh, in improvement. And there are reports that better forms of polyploid have been obtained. Further, polyploidy is associated with increased fruit size, larger plant height or growth, and bigger leaf size. Individually and collectively, these parameters are of high significance. Again, I say, especially in those fruit crops where the fruit size is relatively small. Never take any crop which is already well established and have genetically acceptable size. Example, if any fruit, say apple pear, is taken and ploidy is attempted and size increases, if not approximately to double the original size, even when 1.5 times, the size would increase, but the fruit would ultimately be thickly with more water content, and if delayed in harvesting, the fruit would not fetch any direct beneficial to the cultivators. These five methods over several thousands of years, they were practiced by the uh, human being when he was nomadic, and now the development of science and in recent times the biotechnological techniques, they have promoting the development in a very fast manner, and changes which otherwise were not available. Say, uh, in nature, the species did uh, pollinate among themselves. Fine. But what was the ultimate result? Seed that was formed, and by chance, through artificial method by human being or in nature, the seed was dispersed. But since the embryo was not fully developed, and gradually it degenerated and died, meaning that the diversity where two different species, which otherwise is very difficult to cross, got fertilized in nature, but of no ultimate use, as this huge diversity was lost as the seed or embryo aborted at early stages. Now it has become a common practice to breed at species level, make the cross, and rescue the embryo at very early stages before it starts degenerating, and culture under aseptic conditions, and in a way this embryo is rescued, that is, which was dying in nature is now living in laboratory conditions. Once it establishes, then variability obtained is undescribed because such a variation is not present in nature. Once a seedling has been developed, it does possess lot of difficulties. These difficulties are related to their fruit formation, development, growth, and with backcross methods, these hindrances are overcome. Difficult job, but still attainable. What was not being obtained in nature are now being developed through the biotechnological techniques. Uh, Apart from uh, embryo rescue technique, molecular markers have now found their way into the support of crop improvement, and here their role is significant because in a huge country like India, several names are given to a type cultivar species which on migration not at that very place, and people start giving it several names, and in India vernacular names are given, and they confuse the scientist about the exact nature of the plant material. And through molecular markers, it has become important that these misnamed misnominers, they are being rectified as they reveal the exact nature of the genetic material and not the effect of environment on the effect. And most uh, observations and difficulties are being overcome, and the exact number of available cultivars in at a country, they are now being established, so that any material that is to be exchanged is now in a pure form without any vernacular name attached to it. And similarly, it does serve an important purpose, that is, you are sure which plant material is taken for breeding purposes and not by what is being delivered by the local people regarding its performance, because the local people where this genotype, original species, is growing of its own, they are used to their characters. And scientist who is far away from the place of its habitat, in fact, does not know the real nature. Morphological features established in one or two visits, and thereafter through uh, these biotechnological and molecular markers, the improvement has been sufficiently improved, and in times to come, the breeding cycles will be highly reduced, and beneficial effects of these various methods, whether in solitary way or in collective form, would ultimately promote enhancing the variability as well as in the output of new cultivars with several desired features would be obtained. Thank you.
Thank you.
Madam, just a minute.
Okay, sir. Okay.
Okay, madam. uh, well, I would like to share, that is, in the
कितने स्लाइड
primitive.
बनाए थे पहले? अरे कितने स्लाइड बनाए थे सर की? हेलो, अरे कितने स्लाइड बनाए थे कौन सर की? 25 से 25 पूरी इतनी जल्दी? 25 स्लाइड हो
Hello?
गई शायद ना? Thank you.
Hello?
Hello?
शायद मैं देखता हूँ।
आवाज ही नहीं आ रही।
Hello?
Hello?
Yes, madam. Ah,
Hello?
not audible, madam.
Yes, sir, you are also not audible.
Please mute.
आवाज ही नहीं आ रही।
म्यूट कीजिए।
खत्म कर दिया तुमने?
आपको नहीं आ रही?
नहीं, सर को भी नहीं आ रही।
आपको आ रही?
Madam,
हेलो?
madam, madam, mute करिए. Yes, madam. Just for a few minutes. Hello, madam. Hello, madam. Just for a few minutes, I would like to inform the students that is very early times when he was nomadic and he was moving in the wide farms at highly populated forest for to suffice his hunger. And his major tool was the stones by which he attacked the animals. And it happened in a random way, that is, sometimes the man died and sometimes the animal died. And once his near ones died, he felt very bad. And it was happening in nature over years together. And on an occasion, what happened? He had a pointed stone, and when he hampered it to the animal, it pierced the body of the animal, and the animal died. And thereafter, he realized if a raw tool like sharpened edge stone can kill the animal, then he started developing these tools, and he could uh, make prey of those small animals. And gradually he went over large areas where he found diversity comprising of both useful as well as the uh, poisonous nature of the uh, crops. Sometimes again it happened that he ate a fruit which was very poisonous and he ultimately died. Seeing his nearby dying, the people shared again visiting that very type in the jungle. And when the population of the animals increased, and he realized the importance of certain fruit crops, this nomadic man started shifting to lower places where he could find water. And where was the water? The river areas. And close by, he could find caves. He settled there. He acquired the knowledge of burning, and his life became simple. And he could tread the paths where he found that variability of suitable types, collected the material when harvest time was there, brought that material in large quantity, stored in the caves, and the material which was left out was thrown aside the rivers. It is a fact to realize that most of this civilization developed from this rubbish heaps. And in our science, they ultimately provided huge diversity sources. And from here, through various intruders, material was taken through these routes and diversity was spread from one place to another place. Thank you, madam.
Okay, sir. Thank you very much, sir. I owe my sincere thanks to Dr. Giri Sharma, whose presence here today is of great significance for all of us. So, your acceptance for our invitation as a webinar in the Department of Horticulture at DIBNS is graciously acknowledged. I once again thank all the faculty members and students for their patient learning who became the part of this event. I also extend my thanks to technical team of our college and also the head of our technical team, Mr. Sujit, who took painful efforts to make this webinar successful. I am once again thankful to Dr. Girish Sharma for his cordial joining and being with us today in this morning at the Department of Horticulture. Thank you very much, sir.
You- Madam, I am really thankful that an opportunity was provided by our institute to speak on a
Thank you, sir.
subject, and
Thank you,
I
sir.
will
Thank
be
you.
happy. And I will be happy if proposal is submitted so that I can speak to the graduate faculty of your institute.
Okay, okay, sir. Okay, sir. Sure, sir. Thank you, sir.
Thank you.
Okay, sir. Thank you.